All right, so everyone, yeah, Victor entered the Shogun world in last Google Summer of Code, and he did latent variable SVM part in this project, and I think he will show us some application on latent structured output variable SVMs even today.
Ooh, stop. Okay, hi, my name is Victor. I joined Shogun around February. That's the first commit I did. It's a Jansen Shannon kernel. What I did, a small patch, and then I participated in GSOC in 2012. Alex was my mentor, and I was working mostly in latent SVM framework implementation, which was more or less okay. Yeah, I'll show you some demos. If you get really tired, I suppose you already are. I'm just gonna skip through the pages. Basically, what I'm gonna show here is how the whole latent thing started, and then it got into the structural output as well, and then I'll show you some demos. I'll try to be fast as possible. So basically what we have is like we're extending our problem with a latent variable, which is Z in this case, and we generate, so if you said you were looking at the previous presentation, then in case of like you were seeing the phi was generated by X and the structure Y, here Z is a totally free latent variable. And so your minimization problem comes down to this problem, which is, you will see, is a semi-convex, and you will see what semi-convexity means in this case. Note that if you narrow down the size of your latent label of the set, it's basically just a linear SVM. So what you're gonna, why is it linear? This semi-convex is because in case of, it's a binary problem in the first case, and if it's the negative labeling, then it's all good. It's gonna be a convex problem, it's not a problem. But as soon as you have the positive labelings, that this function is going to become concave, and then you're gonna have to do some tricks, and those tricks are done different ways. But like I didn't mention, but there was a reference in the paper, in $1,000 paper, which is basically applying this method into object detection. Basically what you're gonna do is you narrow down your data set, like latent labeling, into just the positive ones, and you use the positives and always evaluate this function at first, which is basically finding you the best latent label with the current W weight vector, and then you optimize the simple SVM as you saw, and then you do this until you find basically when your whole optimization basically stops, or it doesn't get better after a while. Implementation in Shogun, pretty easy. You have the latent SVM as a class. It needs two things. More or less one, basically a latent mobile that will need a latent feature and the latent labels. Why is this abstraction? Is because we don't know anything beforehand on the features. You're gonna one who's gonna define your latent features itself, because it's not just X but Z as well, and the labels same. I'm mixed up, sorry. So the features can be anything, and you don't know how to, you are the one, the user, who's mapping, creating the phi. And you have to give basically the dimension of phi. This is helping me to create the W basically, and then you still have to give this argmax function because of the inference of your latent label. So then let's jump straight to latent SOSVM. This is how it's defined by this paper, which is a student of Joachim's. SVM light everybody knows, I guess. And basically what you see here is this an unconstrained primary objective. And this is basically two concave function with subtracted each other. As you see, this Y is the structural label, just in previous case, and then it got extended with the latent label as well. And problem is that you have to use some kind of a, since this is totally not convex, so you have to use some kind of a procedure to find to optimize it basically. And then one way of doing this, the convex convex procedure, it's here. I will not go into it. Basically, as you saw here, this is F in the next slide, and this is G. And these are, as you can see, these are all both function of W, of your weight. And you go through this procedure, and then you construct your final W. It's like an iterative procedure until an epsilon is met. What you're gonna have here a problem as well. You have to find the most optimal Z at the moment with the given W, WT, and then you're gonna construct this V, and then with constructing this V, you can construct the next step, and it evolves in the next step you basically solve just a simple structural SVM. So how did we do this in Shogun? Well, it's a hack because of Swig. So we have latent models and we have a structural model, and we couldn't find a way to merge these two problems, or we were busy enough to not to find a problem and not to find a solution for that. But basically what we are aiming now, maybe who's working on vector graph, right? We had a discussion a week ago or two that maybe he will finally manage to merge these two for us. So basically what you're going to do is you're going to set your delta function, you're going to find this most violating constraint function, and the usual, as you see in latent model, like so dimension, 5 vector, etc., etc. And so I created a proxy class, which is stupid, but it's like the problem is that I cannot, because of Swig, I can have multiple inheritance. So I'm going to have like this inherits from latent model, and this inherits from structural model, and then these are like mapped together. And well, you don't really care about this if you're just using it, so don't worry about too much. Problems. So we're gonna have a lot of problems because of these that are what is it? Features, labels, and the model is all basically abstract. You have to implement it, and there was no way we found out to do a director class to be able to feed into each other these classes. So I hope to have a better redesign of this or refactoring the code, but at the moment, or at that moment, we couldn't see any possible ways to fix this. So anything you want to do with this, you have to do it in C++, I'm sorry. Or I guess the static would do it, I guess. So, and then the proxy class is a big problem. But the nice thing is you can feed anything basically, anything that you have implemented in Shogun as a SVM solver. So what we have there is these three. These are bundling methods, and the nicest one is this one that got implemented somewhere around last October, or yeah, somewhere there. It's basically a non-convex bundle method, so your optimization problem can be concave, and you don't have to do the tricks with the CCPP, anything, it will straight away solve your problem. It's pretty good with the results. This is basically just a copy-paste implementation in Shogun of Joachim's implementation. What he gave away, I think it was called SVM struct plus the latent stuff he gave, and the primal mosaic you can feed into it as well. So it's no biggie. I'm not going to go into details. Basically, we give a very simple but complicated way of doing on a picture a structure of a graph. So, and then we optimize it basically. So what we have, in my case, is that we have a radiograph of a chest, and because I was working in medical image analysis, and then with this radiograph, what I aim to solve the problem is that you have the ground truth. Some doctors did the segmentation, and basically I said that, okay, I want to detect the right lung. Yes, it's a director seats because doctors say right lung is a left lung. So I want to detect this in the images. You'll see later why. And basically I defined a very easy graph where I had the root node always in the centroid of this area. The lung, the right lung, and the uppermost and the bottommost part. And then I fed it into the optimization problem. You have to generate, of course, the hawk features, and basically this is what you get. So this is your model. You might have seen it in the Pascal challenges, like the latent SVM is generating this as well. And then you basically run it, and then you do basically a convolution of this model into your image. And this kind of like assures you scaling variance in a way if you do a pyramid of images, and then you end up having these bounding boxes. I don't know if you see the red one and the blue one. The blue one is the correct one, the red one is our prediction. And you have a Jaccard index of 70%, but you don't care about this because we didn't really want to have like a fine grade segmentation of the lung. We just wanted to find it within pictures. And then here you see examples, but let's better switch it here. So basically, here you see a merge of two open source codes. So one of them is available is this code. It's done by some OpenCV people, and basically do you all the inference, and you have the MATLAB code if you want of training. But you can do this in Shogun as well. It's faster, so why not? So that is available as well. And basically, I pre-trained you for some models, and there you see an example coming here. Yeah, can you see it at all? I might be able to resize it. Yeah. So this is the nice part, where these are what I'm going to show now here. It's mostly grabbed from Google, so it's an image search put out for X-ray chest lung. And this image, like a compound, this is like, I had a previous problem where I had a problem with compound images. So basically you had like various modalities, and what you wanted to have is to detect the right lung inside this. And you have two right X-ray chest images here, but the bottom is this is a CT, and this is some 3D, I would say something that inferred from a ultrasound. Yeah, something like that. So basically it detects your right lung roughly, rough enough, but it's good enough for me because you'll see the later ones. Okay, here's an example from, okay, sorry, this happened and I don't know why. Yep, here's an example for the data set as a train test set. You see it finds the right lung pretty much. You don't see, sorry. Maybe now. And you have the root node, two children node, that's it, you're all good. And let's see one more. Yeah, you saw this one. Okay, I prepared something like, yeah, and this could be interesting as well. Nope, sorry. It's not the data, but OpenCV is giving me some buffering problems, or Qt actually. So this as well, it's a small one, so the resolution was quite small compared to, like, it was 1/4 what I used for training, and it works pretty well. But yeah, let's go to the next problem. You'll see this later. Where's my next previous slide? Did I exit? Yeah, I think so. Okay, so the next problem is, yep, was this. So we had like histopathological images. So basically this is a mouse brain with some genome expressions, used some chemicals, and then you have some genome expressions in it. And then basically what they wanted to find, these are huge images, 14,000 times 5,000. And yeah, I'm sorry, but I'm really idiot in this part. So what they say, they want to find the hippocampus, and that the hippocampus is here, and you want to segment this, and you want to measure it the size because it's a good early biomarker for Alzheimer's disease, for instance. So yeah, this is the train set. It's pretty nice that I can say that like we used 5 images and 11 parts defined on it, and then we tested on 20 different images and it worked pretty good. Basically what we said is like, we want to find it, we want to see if really it finds this, but I think I can do it. Nope, sorry. So here's your 11 parts. And nothing else, basically. And then we go back. This is zoomed in. It's a cropped one. And then on top of this, we created like a quite complex, using this pre-knowledge of localization. So we localize the image, the object or the organ, whatever you call it. And then using this localization, you at that point, and those parts you saw earlier here, we use a fine-grained segmentation like level set, but you could use any kind of segmentation. And on that part, you do only the segmentation. You don't care about the rest. So you discard all these. And trust me, this is like, I would say, more than 80% of the thing, of the whole image. So you just use it here, and you do your segmentation there. And we were hoping that you get a much better result. And basically, it's a level set that is driven by different terms or features, you could say. And these features are fed into a classifier. And that classifier is basically pushing the level set to the right direction. And then here you see, this is how you initialize basically your level set. And that it will grow or shrink. And then I prepared here another demo, which should work, hopefully. And if it's not, then I have to restart it, and you have to wait a little bit. So basically, yeah. So it's going to grow the tree. So our problem, or our finding, it's that any of the SVM was not good enough. So we use structural latent SVM to train the localization, but then I said like we are using using classifiers to drive the level set, and the classification using SVM was not good enough. We put it in a simple binary random forest, and it was like magic. It was perfect. So, and that's what we really miss in Shogun at the moment. I'm trying to work on that as well. So some decision tree-based classification, we are totally not having that. Some were implemented by somebody. I don't remember exactly who was that. Some very, very exotic decision trees, and yeah, this is going to go up to 200, so you're going to have to wait a little bit. And then you'll see the actual classification of in this window of the lungs using that. So this is pretty much what I wanted to talk about it. I'm going to still grow the tree, and then in the meanwhile, you can already ask me questions. None? Yeah, I'm going to ask in the IRC as well. Yeah, go ahead.
I'm curious about your random forest stuff. Any ideas why that worked so well? I mean, I guess you tested a few things in Shogun as well.
The biggest problem was actually not really the accuracy, because when I was using a lot of preprocessors and scaling right away the features, it was all good. But random forest was just like tons faster. And I put in a lot of features, so we used a lot of features, and that was as well, you know, it kind of like makes sense why it was faster.
I didn't understand. I missed, I guess, one part is what was the latent part in these images? I missed that, so maybe you can
the parts. So where are the parts? The actual parts. So this is where is your actual parts, or like in the simple case, in the structural, not the structural latent part, but like the latent part itself was as well like you have defined a deformable part, and those deformable parts are basically latent variables.
Okay, and these
hogs
boxes.
Yeah.
This was one part.
Yes, that was one part.
These were the top. These were the top end parts, or something?
Yeah, yeah.
Oh, I see. Okay, good.
So, how kind of homogeneous does a set of structures have to be for you to be able to use these this method for isolating them? Like I noticed, right? Like I noticed you're doing the hippocampus there on that set of images. Would you be able to use that same sort of process to segment out other structures within the brain? We had two problems there. So one problem was basically that data set. So we're using 2D. Everything is here 2D. So we didn't do in 3D, but the extension, we checked it. It's possible the localization can be done, can be extended to 3D, and the level set, there's already 3D extension of it. So basically, we had another data set, but that was too few examples. But that was actually a brain
problem. And but there we suspected that the features we are using, so hog, were not just meant to do that. So maybe using LBP as a better textural feature would have been better. But cool. Yeah, our main concern there was that there was not enough training data. I mean, not enough data at all. But you could do it, you could test it, and
cool.
Okay, then I have to demo you something else because this is pretty slow. So basically, yeah, this is going to be open source as well as soon as we manage to do some. Either we publish it or we don't, and then it's going to be open source anyhow. So just let me fix it for you. Then you see the learning part as a GUI, and that's good enough, and that's pretty fast. So here you see all the, so we offline already extract the features, so we don't have to, on the fly, extract them for all the images, and then using these features, as I said, we use the training and doing the level set, and then you'll see now here soon in the GUI what's happening, and pretty much the same in case of training, testing as well. Slowly, and we are there. Come on. Yeah, so you see the two lungs here. I don't know if you see it. Can you see it? No, I cannot resize this. I'm sorry. Yeah, I can do it. Yes. You see it? So basically, you have the lungs, both of the lungs, you do the localization, and then you grow into those regions. And that's what's going to happen on the next part as well, on the testing part. But then again, yeah, we tried with the, so we tried with the mouse brain. I'm just showing this because this is the data set I had at hand. And with the mouse brain, it was like, the good thing with this whole setup was that we didn't have to fine tune the terms, the parameters, etc., etc. We used the one set terms we find it's good enough for everything, and it's currently performing the state of the art, or even better. So
I'm just interested from, I don't know, I've done some like fMRI sort of, or MRI stuff with human brains, and there's processes out there for doing tissue segmentation.
Yeah.
It's just not very good, and it requires a lot of cleanup work afterwards.
Yeah.
They get close, but there's always spillover.
Yes. I mean, that's the problem with the whole thing. That and then that whole idea came for me actually is that like you have a full body scan and you have like perfect segmenters, let's say, so-called ideal segmenters for lungs, but it's not the same for your liver, or vice versa. And then if you use the localization, then you just run it on the whole body scan. It localizes and labels your organs, and then you know by hand that your segmenter, whatever segmenter is the best, and you choose out that one from the pool and you run it because you know what's in there. And you already know the location, so you don't have to run it, like say your liver segmenter on the brain or the whole image because it's useless.
Yeah, cool.
Yeah, you can watch it. But that's all. Nobody have any questions in the IRC either. Thank you very much.